Here we are in the Raman Laboratory of the Department of Chemical Sciences. This was the first Raman Laboratory here in Padua, at the university. Raman Spectroscopy was an important spectroscopy. Started in 1930 when there was the Nobel Prize for Raman, an Indian scientist. And then it was developed with the laser. We are using laser now for exciting sources. And these lasers are started in the 60s. And so here in this laboratory, it was dedicated to optical spectroscopy. Raman started at the end of 60s and beginning of 70s. Raman spectroscopy is used in many applications. It is now a very, very important spectroscopy used in material science, in medicine, in a lot of other fields, and also in particular in cultural heritage. In this period, it is also used with artificial intelligence, and so there is a lot of work that we are doing also with Raman spectroscopy.
Raman is specifically very useful for the research in cultural heritage because of the possibility to have a very high spatial resolution. So it's specifically indicated for the characterization of pigments, dyes, and binders, for instance, so materials you can normally find on painted surfaces. And thanks to the power of Raman, which is one of the techniques more able to identify in a reliable way the materials, Raman is widely used also on site. For instance, we have also portable Raman instruments which are very used on site for the characterization in museums, in libraries, and also on archaeological sites. And is specifically useful when you can cross the Raman data with other data, as in our research, like micro-FTIR or X-ray fluorescence data. And the crossing of these techniques provide generally quite a reliable picture of the materials you have on your artifact, even if you have just a very, very small amount of sample, like in our cases where we worked with really very, very small amount of fibers and materials.
The specific research topic arose from the study of some microlithic tools found in Caucasus region dated to Upper Palaeolithic. So with a colleague of Venice, we want to identify this kind of micro-residues, blue-dyed, found on the active area of these tools. And so due to the tiny amount of material of micrometric size plant residues, we decided to use some vibrational spectroscopy with micrometric resolution to identify both the dye and the vegetal residues on which this dye adhering to. So with the Raman here, we identify the dye. We understand it is indigotin, which is derived from the processing of Isatis tinctoria leaves, whereas the vegetal residues was bust-like residues relying on the lignin and the pectin features, diagnostic features.
This is a micro-FTIR Lumos 2 instrument, which is a recent acquisition under the world-class research infrastructure Sicuri. We used in this research these instruments to better characterize the fiber materials and the organic components present inside the materials we have analyzed.
So Raman spectroscopy was used in this case for identify these residues and these samples from 34,000 years ago. But it is used nowadays for many other applications. We use, for example, it for nanomedicine, in which we are able to identify antigen on tumor cells. So there is a wide possibilities of using this Raman spectroscopy, because also the resolution is very high since we are using this microscope, and so the resolution can be down to the order of 1 micron. Namely, cells, for example, are a dimension of 10, 20 microns, so we are able to see inside each cell. So applications are many, and we think that we will develop further in the future this type of spectroscopy.